So you are going to be receiving, or hopefully you're picking up yourself, the kit for all the experiments you're doing with the laboratory portion of your General Biology 1 course. This is for currently the summer 2024 section of the course. If you find an older one online, it's not going to be the same. There are a bunch of new things that we're doing this year that aren't included in the old one. So what I'm going to do is I'm going to go through a list of the lab work that you're going to do, the experiments you're going to set up. Not every detail for each experiment. There's separate instructions for every single experiment, how to set it up, what kind of data you're going to collect, what sort of calculations you're going to have to do, what sort of how you're going to interpret data or present that data, and then do research on answering questions related to that experiment. Some of these will take a short amount of time to do, others will take several days to do. You're going to actually have to set it up and be collecting information over a several day period in order to complete all the work related to that lab. So you need to have your kit ready to go and you need to start doing the work as soon as possible. You will not have enough time to finish it if you let these things go toward the end of the class. You can't do it in a week, it's not possible. You have to, most of these things will take like 4 weeks minimum if you start right from the very beginning. Some of the things will take that long in order for you to do. So you need to get on this as fast as possible. What I'm doing is I'm going to pull out material from the kit and talk about which labs they go with, okay? So the first lab that you're going to see, it's not really even a lab, it's just kind of a worksheet on what is a microscope, how do microscopes work. That doesn't have necessarily a practical portion to it. It relates to work that you are going to do in the lab with a microscope. And how it works, magnification, and a variety of other things. So you need to kind of have the background, right? But that's a separate thing. The first actual experiment you're setting up is the effect of sugar concentration on yeast cells, right? So there is an explanation online of how to do that. You're going to need a test tube rack, right, to set that up. You're going to need 50 mil centrifuge tubes. Now what you're going to find is that inside some of these 50 mil centrifuge tubes are other things, like so your counting slide, the hemocytometer, the slide that's going to be used to count the yeast cells is actually in one of these. So you're going to need to take it out. There's some extra slides and cover slips in there. This one here has some little bit of blue dye. Put that right here in your test tube rack. The yeast that you're going to be using in this yeast experiment here in another tube. Okay, so that's the second one here. So you can see, you can just have a little place for those to go. And then here's this one, doesn't have anything in it. So you're going to need these 3 tubes for this first experiment. You're going to need the test tube rack. You'll need the yeast. You'll need the dye. You'll need a pipette. There's a little place you can put the pipette, right in here too. So this you can get this test tube rack. I think that's mostly for getting the first one set up. You're going to then I have an explanation of this, adding yeast and water and the dye, and then you're going to be using the microscope. You're going to be using you may get a different version of this, okay? So this is like a little stage for the microscope. When you put the microscope on this, it's like over the mirror, and it's going to reflect light back up to the lens, and then it'll really help in the contrast from when you're looking at the counting slide. So I have a new version of this that I'm currently making where you can adjust the mirror's angle and the angle of the light and the contrast. So it's pretty good, but I'm designing and building it in a CAD program, and so you may not have it in your class. You may have this simple version. Okay, so you should have those sorts of things in the kit. And that's mostly for this first lab. Second lab is calibrating a pipette. So you're going to get a digital balance. This digital balance is pretty good. It goes to 2 decimal places. I've compared it to balances that we have in our labs that cost several $100s, $300 to $500. This is equally as accurate as those. It is slower, so you go to weigh something on it, you got to wait a little bit, but the accuracy is the same. All right, you're going to have a weigh boat. You're going to use this in many, many labs. You're going to weigh all sorts of things, liquids and solutes. So you're going to do that. Some of these things have batteries, so you may have the batteries provided for you with it, and others you may have to get some batteries. Okay, so you'll have a little shopping list of your own. So for calibrating a pipette, you're going to just have your pipettes. You're going to need some water, and you're going to have the digital balance and the weigh boat, and you're going to be kind of pipetting liquid water into here and then kind of marking on the pipette with a Sharpie, which is not included in the kit, but up here you're going to need a permanent marker of some sort to then mark on your pipette exact volumes. Okay, so you're going to just calibrate the pipette. After you calibrate the pipette, there's a second in lab where you're going to then have different volumes. I'm telling you which volumes you're selecting, and then you're going to be pipetting it there and then just looking at the weight. You're not adding it till you get to a certain weight. You're reading it on the pipette, then dispensing it, then seeing how much does it weigh, and it may be more and it may be less. That's the point. You're trying to get the same every time, but you're going to record data, numbers from the balance, and then you're going to use that to calculate means and standard deviations. You're going to do different volumes, smaller volumes with this one and a little bit larger volumes with this one. And then you'll be using these in future labs, so this way you'll be oriented to them as well. The next one, you're going to be doing serial dilution. So then we will be replacing these bigger tubes with several of these smaller tubes. Okay, so we'll be putting these other tubes in the rack. You can use standard food coloring in this, or you should have plenty of your dye from the first experiment left over, so you could use that. You're going to be doing a worksheet on planning out the dilutions, and then you're going to be physically making the dilutions. So you'll be reading the volumes on these little containers, and then you'll be pipetting the dye, and you'll be pipetting liquid from one to the next. So again, there's a whole explanation of that, but you should have a whole series of these. Actually, there's quite a bunch extra. Same thing is look at them, because they will probably have something inside them. Before you start adding liquid to it, make sure there's not something inside the tube. All right, and many of them will be reused for multiple experiments, using the same thing over again. There's a standard curve one where then after doing this series of dilutions, we could read it on a spectrophotometer, which you don't have. So then you're given data and you're going to graph. So you're going to be doing graphing in an Excel or a Google Sheets program, and then you're going to be creating graphs to get equations for a line. You're going to do that 3 times throughout the course, progressively. One, just create one. Second one is create one, then to use it to solve for unknowns. The third one, you're going to create one, use it to solve for unknowns, and then use those calculations to do other calculations to find out a rate of reaction. So it's going to build. So this is a specific type of skill that you're going to have to do with working with data to generate standard curves. So that's kind of related to the dilution one, because that's where the data for that comes from. Then you're going to do some molar solutions. So now you're going to go back to needing your balance and your Weibo. For this particular one, what you're going to have to do is once you do your worksheet, you're also going to make a dilution. Now, I'm not going to be able to see exactly whether you made it correctly or not in person, but what you're going to do is then photograph your solute that is weighed on the balance with the weight of it, and then you're going to measure out the volume on this graduated centrifuge tube, and then you can take a picture of that. So kind of take a picture of both of them together. And then when we look at that, we'll say, okay, yeah, you actually did it and you did it right. But if I look at it and I see the volume is wrong because you didn't read this properly, then I'll let you know, and you might lose points for not measuring these things out or doing the calculations properly. Okay, but this is a way we can check this in person. Everything you do is going to require photographic evidence. You can only get half of the credit for all the work you do if you only provide worksheets and turning in, written information. You have to turn in photographic information of the work that you did, or you cannot get full credit. You only get half the credit, right, for the different types of lab work and experiments. So you're going to do a whole bunch of problems, but then you actually have to make the solution. Osmosis then is going to continue. You're going to do more molar solutions. So you're going to have to use your balance and the Weibo again. You're going to have to get sugar on your own, measure out the sugar, make these different solutions. You should have then little cups like this where you can kind of put the different concentrations in these cups that you're going to have. And then in one of your other tubes here, you're going to get a bunch of dialysis tubes. So the dialysis tubing looks like a little piece of plastic. Okay, actually there's a bunch of little tiny holes right in this. And what's going to happen is you're going to turn this into like a little cell. So there are rubber bands in here as well. And I'm going to show you in another video how to do this. You're going to get this wet, take one end, seal it, cut the rubber band, tie the rubber band around it, then take your solution from here, that's a sugar solution, use your pipette, you're going to pipette it in there. I'll tell you the specific details, the volume. You're going to then seal it, and then you're going to need beakers. You're going to need, or it doesn't matter what they are, just drinking glasses. And there's going to have pure water in them. And that's it. And then you're going to drop these into it, and these are just going to be bags full of sugar. So it's nothing harmful about it. It's just completely, household, kitchen kind of type things. But before you drop it in the water, you're going to weigh it on your balance. And then after it sits for a while, you're going to take them out of the water and then reweigh them. And then put them back in the water and let it go longer and then reweigh them again. And you're going to have a whole bunch of different sugar concentrations because you're going to be making these molar solutions. And then we're going to compare those. We're going to say, what effect does the molar concentration of the sugar have on the rate of osmosis? And you're going to be doing that with dialysis tubing and water and all these things. So everything you need will just be here in one of these little containers that says dialysis tubing. So don't lose it. It's very easy to miss because it's transparent and you can't actually see it. So that's an osmosis lab. Then you're going to look at the effect of light on cell division and microalgae. So I'm still actually setting those up right now, but you're going to get a little tube like this with some live algae in it, a Euglena culture. And then what you're going to do is use some tubes like this. You'll actually just have 3 of them. One of them is going to be 24 hours of light. So you're going to need like a little desk lamp or something that could be on 24 hours a day. One of them is going to be completely in the dark. So in your kit, you should have aluminum foil like this. And what you're going to use the aluminum foil for is covering up the tube. Now, you're going to need enough of this to cover 2 tubes. So it's enough to cover 1 tube, tear it, and you're going to need that on a second tube. One tube's going to be all dark, another tube's going to be all light. This tube will get about a 24/7 light cycle, so you'll cover it about half the day, and then at some point make note each day when you're going to uncover it. So it will get light for 12 hours, be dark for 12 hours, and then you're going to be using then your microscope again, your stage, and the counting slide, which is inside here right now. I'm going to pull it out just so you can kind of see. There are a bunch of slides in here. The counting slide, all right, like this. It has 2 chambers, and there's another video explaining exactly how to use that, but it's where you're going to be looking at the cells and then actually counting them because there's a little grid on the slide. So that's going to be setting up the algal experiment with those extra tubes that you have. That goes for several days, so you have to get it started early on because you're going to kind of count the cells and let it go, and count the cells again, and let it go, and count the cells again. So you're going to keep coming back to it. You have to keep cleaning your slide and allow that culture to go for a while. So it'll be in the instructions. The next one on this list is calculating protein concentration with a standard curve. So that's what I already talked about. You'll be given data, you create a graph, you do a bunch of calculations, you figure out the concentrations of unknowns with that. So there's nothing in person you're going to do for that particular one. That's more with data that you're given. Now you're going to look at the effect of enzyme concentration on a reaction. So you're going to have to get a potato. You have to blend up the potato with a blender. If you don't have a blender, you could just smash the potato. I could show you in a video. You're going to need the little medicine cups. You're then going to use, this is just coffee filter. You just need one at first. Ideally, what you do is you'll just fold it like this to create like a little funnel. See, and then you're going to be pouring your blended up potato through this, collect it in just a drinking glass or a mug, and then you're going to take that and you're going to perform a series of dilutions in these cups. So you'll have a bunch of diluted potato juice, but it's going to contain an enzyme. And then in one of these tubes here, find this one, there are these tiny little, you can see something white like flipping back and forth. They're little filter paper discs, so little circles cut out of filter paper. And what you're going to do is, using the forceps in your kit, you're going to add a certain number of these into each of the solutions. Then you're going to get one of these containers, set that up, and you're going to get hydrogen peroxide. So for this lab, you're going to have to buy a few things. You're going to need the potato, and you're going to need hydrogen peroxide. You're going to put your hydrogen peroxide in here, and then you're going to take little discs out with the forceps, and then drop them into the hydrogen peroxide. If a reaction occurs, oxygen bubbles will form and the little disc will float, and you're going to be timing that, how quick it happens at the different concentrations. Right? So you're going to just need the little discs, the forceps, these medicine cups, and your centrifuge tube for that lab. You'll have a lab where you're going to measure the pH and adjust the pH of some things. So you're going to have brand new. We just got these pH test strips. We tested them out. They're fairly accurate. And a little color chart you're going to be able to use to compare different solutions and see. I'm going to give you on your worksheet a list of household things I want you to go find and then measure the pH of, and then you're going to measure the pH of some soil by taking some sample in one of these tubes, mixing it with water, and measuring the pH, and then you're going to add something, baking soda or lemon or lime juice. You're going to have something that might then alter the pH, and then you're going to test it again and kind of see what effect that has. So there's a bunch of instructions with that particular lab, but you'll be looking at pH factors there. The next bit of work that's done then is the effect of pH on enzyme rate of reactions. So these things kind of fit together. You did an enzyme lab, then you did something with pH. Now you're going to look at the effect of pH on activity of an enzyme, except this is a little more complicated lab. So for this one, you're again just given the data from it. You're going to have to make some graphs, do a whole bunch of calculations. There's actually a lot of work to do with this one. You're not physically doing this particular lab with the tyrosinase, but you have a lot of graphing to do, calculations to do, and research. So this is one of the more time-consuming ones, even though it's not necessarily a hands-on one. There's going to be some plant pigment chromatography. So you are given here a tube that is going to have solvent in it, and then you're given here a bunch of chromatography paper, all right? And then you're going to have to get some plant material, and you are going to then put pigments on the paper, put the chromatography solution in here, let it move the pigments, and then you're going to measure pigments. There's again the whole explanation there, but and then you're going to try it with some pigments of your own. You'll be doing that inside this chamber, so that'll be the chromatography chamber for you. And then the last two experiments that you're going to do, one is with mitosis. That is a bunch of pictures of mitotic cells taken from a microscope, and you're going to identify them and then count them in the different stages. So you're going to be looking at that. And then also there's going to be genetics problems that you're going to be working out, which are going to be, again, mostly from worksheets that you're going to do. So your kit is going to have, a lot of things that you're going to need to use multiple times. A lot of these tubes you're going to use over again many times. The microscope you're going to use many times. You need this test tube rack to hold stuff for all the different labs and your pipettes and everything else. So everything you have, it should be in this, everything that we're going to provide you here is in this kit. This is one that's actually I just pulled off the shelf, so it's one that a student would have. But there will be some things that you need, and that's going to be a little separate explanation for your shopping list. So what is it that you have to go out and get? Like I mentioned, a few things like the potato and the hydrogen peroxide and a few other things. So some really simple, inexpensive things. You may just have them in your house. You don't even have to go out and get them. But if you don't, there are a few things you'll need to complete all the labs in the class.